And now there are a couple of topics that we really do want to consider and cover in this chapter, but I want to do it in office hours because the concept of low barrier H-bonds takes a little bit of back and forth and an interactive approach, and I want to do that, as I say, in office hours, so we'll get to that. So we'll move on for a little bit here. And I also want to talk about quantum mechanical tunneling, but that's another rather interactive discussion that we should have. So let's put that off for office hours. And then we'll talk about metal ion catalysis very briefly here, just making the point that enzymes often have metal ions that can accelerate reactions. And in this particular case, you're looking at thermolysin, the active site of thermolysin, which is an endoprotease that cleaves peptide bonds. And in this diagram, you can see that the zinc simply stabilizes a negative charge on the tetrahedral oxyanion in the middle of the reaction. So we're going to come back to that. We're going to see metal ion effects later on in metabolism, so we won't spend too much time here. There's a wonderful discussion here of antibiotic resistance by superbugs. We're going to leave this to your own reading. You might want to read this for the MCATs and so on, but we won't discuss it here. It's a wonderful story, and it's very timely because it's in the news a lot, but we won't talk about it here. And similarly, we won't say much about how active site residues interact to support catalysis. I think you can not cover this in your own reading and your own study, so we'll move on to another topic. And there's also an interesting box in this chapter about how electric fields can accelerate enzyme reactions, and it involves something called the vibrational Stark effect, and the work of a biochemist by the name of Stephen Boxer. We're not going to cover this either. I don't think you need this for the MCATs, and so we won't talk about this, but it's a really interesting story in case you're interested. We want to move on and talk about real enzyme mechanisms now, and our first example is going to be the serine proteases. And you actually ran into these way back in chapter 5 when we talked about trypsin and chymotrypsin and the reactions they catalyze. And it's important to remember that proteases are enzymes that cleave other proteins. They cleave peptide chains. And so the reaction we're going to see is a reaction that cleaves a peptide chain by breaking the amide bond in the middle of a peptide. And chymotrypsin and trypsin, of course, are endoproteases, so they cleave in the middle of a chain. And they also happen to involve a serine in the active site of the enzyme, so that's why they're called the serine proteases. And in fact, that serine is involved in a covalent bond in the mechanism of the enzyme. And so we point out that the enzyme and the substrate become linked by a covalent bond at one or more points in the reaction pathway. And the formation of the covalent bond provides chemistry that accelerates the reaction. Serine proteases also employ general acid-base catalysis, so we're going to emphasize the point here that the serine proteases use both covalent catalysis and general acid-base catalysis in their mechanisms. And we'll talk about that more as we go along. Now, typical examples of the serine proteases include trypsin and chymotrypsin, and elastase, which was discussed in chapter 6 in the story about alpha-1 antitrypsin. Thrombin is a blood clotting enzyme. Cetylysin is a very tough protease that's actually used for contact lens cleaning solutions and so on. And plasmin is involved in breaking up blood clots. And TPA is tissue plasminogen activator, which is known as a clot-busting drug. It's actually used to break up blood clots in cases of people that have had heart attacks and strokes. We may come back to that and talk about it later. All of these involve a serine in catalysis, and that's where the name comes from. That serine residue is part of what's called a catalytic triad of a serine residue, a histidine residue, and an aspartate. And we're going to talk about the individual roles of each of these residues at the active site. Now, all the serine proteases are homologous. They're similar in sequence and structure, but the actual locations of the three crucial residues differs a little bit from one enzyme to the other. And enzyme biochemists, enzymologists, don't really like that very much, so they've agreed to number these particular important residues in every enzyme in which they're studied as histidine 57, aspartate 102, and serine 195. Those are the numbers based on chymotrypsin. And so you have to sort of renumber the sequence of the protein in some of the other enzymes in order to have the histidine always come out at residue 57 and so on. We're also going to mention burst kinetics. Burst kinetics provide evidence for the involvement of a covalent intermediate in an enzyme reaction, so it's very important that you understand that. Now here's a picture of the serine proteases and the sequences of chymotrypsinogen, the precursor to chymotrypsin, and trypsinogen, which is the precursor to trypsin, and elastase. And you can see where the important residues are located in each of these cases, the histidine, the aspartate, and the serine. Here's a picture of chymotrypsin, a nice structure showing chymotrypsin in a complex with eglin, a model substrate. The chymotrypsin is in the beige or brown, and the eglin is the blue or sort of teal-colored protein molecule in the structure. You can see the three critical residues. The histidine 57 is in red, the aspartate 102 is in gold, and the serine 195 is in green. We're going to talk about what each one of these residues does in the mechanism. This is another way of looking at those three important residues. Shows the interaction of the aspartate in the lower left here with the hydrogen bond between the aspartate carboxyl and the hydrogen of the histidine in the active site, and the other nitrogen in the histidine is in a hydrogen bond with the OH of the serine 195. So that's another way of looking at this, and we're going to see other pictures of the mechanism and of the active site as we go. This is a picture of the binding pockets of the three enzymes, and it explains the specificity of trypsin and chymotrypsin and elastase. We won't talk about this directly here, but we did actually discuss this way back in Chapter 5 when you learned that trypsin will cleave at arginine and lysine residues, whereas chymotrypsin cleaves at aromatic residues. Here's another look at paranitrophenylacetate. It's an ester, but chymotrypsin cleaves simple esters, in fact, in addition to peptide bonds. I'm going to use paranitrophenylacetate to demonstrate burst kinetics for you. In the upper diagram here, when you do a chymotrypsin reaction with paranitrophenylacetate, you see an immediate burst of the first product, paranitrophenolate, and then a slower release with time. And on the other hand, the other product of the reaction, the acetate, shows a lag of production and then a slow and steady rise like this. What's going on? Let's take a look at the mechanism and we can sort this out. Here's the serine hydroxyl in the active site of the chymotrypsin. Here's the paranitrophenylacetate. And the first step in the reaction is the release of the paranitrophenolate, which leaves a covalent enzyme substrate intermediate. When the serine hydroxyl attacks this carbonyl carbon, it forms this covalent bond between the acetate and the serine, and it releases the paranitrophenolate as the first product. So here's the release of the first product, and the acetate doesn't appear in the plot above because it's bound to the enzyme in a covalent complex. Then water attack on the carbonyl carbon here releases the acetate in a second slower step. And so what you're seeing here is rapid release of the first product in the fast step and slower release here of the acetate in the second slower step. That's a demonstration of burst kinetics, and it's evidence for the formation of a covalent enzyme substrate intermediate. Now, the mechanism of the serine proteases is really a mixture of covalent and general acid-base catalysis. The aspartate residue has only one very simple job. It only functions to orient the histidine in the active site. The histidine 57, on the other hand, acts both as a general acid and a general base in the reaction. And the serine 195 involves formation of a covalent bond with the peptide to be cleaved. The covalent bond formation turns a trigonal carbon into a tetrahedral carbon, and then the tetrahedral oxyanion intermediate that you'll see in a couple of places in the enzyme mechanism is actually stabilized by the NH groups of glycine 193 and serine 195. Now we're going to look at the mechanism of the serine proteases in some detail, but as we begin, it's important to emphasize that what you want to know is the chemistry. The art with the pretty borders here and so on is less important than understanding the chemistry itself. So be able to write this mechanism without all the decorations and so on. The chemistry is what's really important. The first step is binding of the substrate. So this is a peptide. This is a protein that is the substrate for the serine proteases. And the bond we're going to cleave is right here. Okay, that's the amide bond that we're going to cleave. It's an NHCO amide bond, and that's what we're going to be cleaving in the mechanism of the enzyme. Okay, so let's take a look. Here's the first step. As we said before, the aspartate is simply forming an H bond with the histidine to orient the histidine, put it in the right place in the active site. And now the histidine acts as a general base to remove a proton from the serine, and that forms an O-, and that serine O- can attack the carbonyl carbon of the peptide to be cleaved, and that promotes removal of electrons from the double bond up to the oxygen to form a tetrahedral oxyanion. In the second step of the reaction, we have to protonate the nitrogen here in order to cleave the amide bond later. Notice that this nitrogen is uncharged. It has a pair of electrons here. It could not accept the electrons from this bond that's going to be cleaved unless we protonate it. And so the second step of the mechanism is protonation of the nitrogen of the peptide substrate, and that occurs when the nitrogen picks off this proton from the histidine, and the histidine is acting as a general acid in this case. It's donating a proton. In the third step now, we have a protonated nitrogen, and the oxyanion here is going to collapse to reform a double bond, and that drives electrons from the peptide bond over to the nitrogen. And emphasize that it's only because of the presence of the positive charge that these electrons can be transferred to the nitrogen to accomplish the bond breaking. In this step, the first product dissociates. This is half of the peptide substrate being released by the protein. The other half is here, and it's remaining in this covalent complex between the serine on the enzyme with the rest of the peptide that is the substrate for the reaction. In this step, the histidine is acting again as a general base, taking a proton from a water molecule this time, producing an OH- right in the active site, and that OH- attacks the carbonyl carbon of the covalent bond between the serine and the remaining portion of the substrate. In the next step, the oxyanion here collapses to reform a carbonyl, and this bond breaks because the electrons are used to pick up a proton from the histidine. Once again, the histidine is acting as a general acid to carry out its part of the reaction, and that leaves us with this, which is really the second product, free now from its covalent bond, and it dissociates and the reaction is over. Okay? There's a little note here about transition state stabilization in the serine proteases. We actually mentioned this before. The oxyanion, the tetrahedral oxyanion, is stabilized by the NH groups of serine 195 and glycine 193. And there's a picture of that here so that you can see that stabilization. Now, the aspartic proteases offer a contrast from the serine proteases because they do not involve a covalent intermediate in their mechanism. Aspartic proteases involve purely general acid-base catalysis. They do not involve a covalent intermediate. For many years, enzymologists thought that the aspartic proteases would show a covalent intermediate like the serine proteases do, but they never found one, and in fact the mechanism is more consistent with pure general acid-base catalysis. Examples of aspartic proteases include pepsin and chymosin in your stomach, cathepsin D in the spleen and liver, renin in your kidneys, and the HIV protease that is part of the AIDS virus and an enzyme that's involved in processing of AIDS virus proteins, and we'll talk about that in just a bit. The mechanism of the aspartic proteases involves two aspartate residues at the active site, and these two aspartate residues actually work together to do general acid-base catalysis in this mechanism. Most aspartic proteases have an N-terminal lobe and a C-terminal lobe with approximate twofold symmetry. On the other hand, the HIV-1 protease is a homodimer, and that's shown here. The HIV-1 protease is a beautiful symmetric structure of two subunits that are identical, forming a very symmetric dimer. On the other hand, pepsin in the lower picture shows you an N-terminal half that's shown in red and the C-terminal half in blue. It doesn't look very symmetric here, but the arrangement of the aspartates in the active site is reasonably symmetric, and that's why we describe it as a pseudo-symmetric active site, in fact. The pH rate profiles for the aspartic proteases are revealing. Pepsin shows a pH optimum of about 2.5, and it was thought for a long time that the rise from low activity to high at low pH represented the deprotonation of one of the aspartic acids, and it was also thought that the decrease in activity at higher pH represented the deprotonation of the other active site aspartate. Turns out it's not quite that simple, and we'll show you the mechanism in just a bit. The HIV-1 protease has a similar pH rate profile, but a different pH optimum of about 4.5. We'll see that the predominant catalytic factor in the aspartic proteases is general acid-base catalysis. The mechanism for the aspartic proteases looks complicated, but we're going to emphasize the similarities between this mechanism and that of the serine proteases. Shown in the first frame are the two carboxyl groups of the two aspartic acid residues in the active site. A water molecule is poised between those two carbonyl oxygens, and in the first step of the reaction, this carboxyl acts as a general base to extract a water proton, and that leaves a hydroxide ion that can attack the carbonyl carbon of the peptide substrate. In the second step, just as with the serine proteases, we have to protonate the nitrogen of the peptide bond to be cleaved, and that's done as shown here. Now we have a protonated nitrogen, and the oxyanion can collapse to form a carbonyl and push the electrons from the peptide bond over to that nitrogen, leaving the two products for the reaction. The reaction's really complete at this point, and all that's left is dissociation of the products. So to review, the carboxyl on the right acts as a general base to extract a proton from a water molecule, facilitating attack by the hydroxide ion on the carbonyl carbon. The second step involves protonation of the nitrogen, and that involves the carboxyl, the same carboxyl, acting as a general acid to donate a proton. And in the next step, the oxyanion intermediate collapses, pushes electrons from the peptide bond over to the nitrogen, and the reaction is done. Studies of the aspartic protease mechanism have shown that there's actually quantum mechanical tunneling going on in the middle of the reaction, going from the ES state, the enzyme substrate complex, to the ET prime state. We'll talk about what those letters and primes and so on mean in office hours, but anyway, tunneling is involved in the mechanism of the enzyme. The aspartic protease involved in infections by HIV is actually a remarkable imitation of mammalian aspartic proteases. It's a very symmetric homodimer, and it's involved in processing AIDS virus polyproteins, as they're called. When the HIV genome is translated into protein, it actually produces what's known as a polyprotein, and that polyprotein has to be cleaved into smaller parts that make up the various proteins of the HIV particle itself. And that processing, that cleavage of the polyprotein, is accomplished by the HIV-1 protease. The structure of the HIV-1 protease is shown here, and the close-up shows the two aspartic residues at the active site. This turns out to be a complex of the protease with the inhibitor known as Crixivan, made by Merck. This is an important AIDS protease drug, as it turns out, that has extended the life of lots and lots of HIV patients. Protease inhibitors like Crixivan are very effective AIDS drugs because if the HIV-1 protease can be selectively inhibited, then new HIV particles can't form. There have been many such inhibitors found to work in a culture dish, but successful AIDS protease drugs have to be able to kill the virus in a human subject without blocking other essential proteases in the body. Shown here are a variety of protease inhibitors that have been very effective in treatment of HIV. We'll turn our attention now to a different enzyme called charismate mutase, and it's really a model for understanding catalytic power and efficiency because of the very simple mechanism that it carries out. The point is that enzymologists have long sought direct comparisons of enzyme-catalyzed reactions and their uncatalyzed counterparts. That turns out to be difficult to do. Charismate mutase has become a model really for making this comparison because of the efforts of dozens and dozens of researchers over the years. The enzyme acts in the biosynthesis of phenylalanine and tyrosine in microorganisms and plants, and involves a very simple reaction of a single substrate and a concerted intramolecular rearrangement that converts charismate to prephenate. In this reaction, one CO bond is broken and one carbon-carbon bond is formed. This slide shows the rearrangement of charismate to prephenate. It's a very simple reaction, and it will remind you of the Claisen rearrangement that you probably know from organic chemistry. It's really the same kind of intramolecular rearrangement chemistry. There are two possible ways that this rearrangement could occur. One of those involves a boat conformation for the charismate substrate, and the other involves a chair arrangement for the charismate itself. Jeremy Knowles at Harvard showed that both the charismate mutase enzyme reaction and the uncatalyzed solution reaction both proceed via a chair conformation, and Knowles synthesized a transition state analog of this structure that's shown here. This shows that same transition state analog in the active site of the enzyme. You can see that the charismate mutase enzyme is a dimeric structure, and the active site requires the presence of both subunits to form. On the left structure here, you see one active site here and one active site here, but both of these involve parts of both of the subunits. And this is the transition state analog in the active site. Here's another look at that same structure showing the electrostatic and H-bond interactions that are involved in the transition state of the reaction itself. This slide shows the postulated transition state itself in the lower right of the diagram. And what's important to realize here is that there are no residues from the enzyme involved in this mechanism. The reaction occurs in the active site, but there are no residues from the enzyme itself that are actively involved in the mechanism. How does the reaction occur? Tom Bruce, who we mentioned before, has shown that charismate mutase really works via a near-attack conformation, and the energetics of the reaction from Bruce's work are shown here. As we pointed out before, the formation of a near-attack complex is very difficult in the absence of an enzyme, and in this case, for the charismate mutase reaction without the enzyme, involves an increase in energy of the substrate by about 34 kilojoules per mole to form the near-attack conformation, and then an additional increase in energy of about 67 kilojoules per mole to form the transition state. On the other hand, in the enzyme substrate complex easily forms the near-attack conformation with a very small increase in energy, and then you have about the same increase in energy from the NAC to the transition state in the enzyme case as you did in the case of the uncatalyzed reaction. Overall, the energy barrier between the enzyme substrate complex and the transition state here, though, is only about 64 kilojoules per mole, whereas for the uncatalyzed reaction, it's approximately 100 kilojoules per mole. So the catalysis by charismate mutase really depends on formation of the near-attack complex on the enzyme itself. What the enzyme is doing is not covalent catalysis, it's not general acid-base catalysis, it's merely formation of the NAC in the active site. The catalytic power of charismate mutase really derives from the easy formation of a near-attack conformation in the active site of the enzyme. That's what the enzyme is doing. It's an incredible demonstration of Bruce's postulate that near-attack conformation formation in an enzyme can be an effective means of catalysis.